I've got hot takes, and some of them are going to make you mad. But listen, science is all about asking questions and rethinking how the world works based on the answers. So a lot of these hot takes challenge what you thought was fact. Like, I know I'm a person, not a fish, and people are supposed to shower every day, and we're supposed to put our medicine in medicine cabinets, unless data suggests that all of those statements are wrong. When data gives you a new way of looking at things, you have to listen. So here are SciShow's hottest takes, starting with a deep cut that's really gonna rile up the die-hard Hank Green fan base. Michael's about to tell you in so many words that butt is not legs.
Why do we have butts? It might seem like a cheeky question, but if you look around the animal kingdom, even at our closest relatives, big butts are pretty uniquely human. So why would evolution grant us such an asset? We have a few hypotheses. So our butts are made primarily of two things: fat and muscle, and each likely has its own evolutionary role to play. Like, in general, fat is a resource that's useful for our brains as they develop. A 2016 study proposed that development for both our large brains and our fat stores coincided with the time our ancestors came down from the trees. See, it takes less energy to transport all that fat when you're walking versus when you're climbing. So by walking on two legs, we could carry more fat, allowing us to develop and power a bigger brain. Or so the hypothesis goes. There's still plenty we don't know about the evolution of fat, and bum fat in particular. It's tough to track because it isn't preserved very well in fossils. However, we're in slightly firmer territory with the muscle part of all this, the gluteus maximus. It's the biggest muscle in our bodies, which is only the case for humans, not our ape relatives. We're also the only primates that habitually walk on two legs, so for nearly 200 years, scientists have thought that these two observations are probably related. Now, your gluteus maximus isn't actually used very much when you walk on flat ground, but it sure is used when you run. As your heel hits the ground with each stride, your body angles forward, and it's that big gluteus maximus that's partly responsible for stabilizing your body and pulling it back. Like fat, muscles aren't usually preserved in fossils, but the bones they attach to are. And the functions of bones and muscles are closely related, so we can make inferences about the evolution of one from the other. We know that of our various ancestors, Homo erectus had a pretty human-looking pelvis around 2 million years ago, which could mean a human-looking butt. This came along with a bunch of other features that make us good runners, things like long Achilles tendons, short toes, and long legs. Humans are the only primates that can run long distances, and scientists think that distance running helped us evolve new strategies for obtaining food, by chasing prey to exhaustion, or by quickly getting to carcasses to scavenge them. So, butts would have been a key advantage for hunting. In support of this idea, the evolution of the butt roughly corresponds to a time anthropologists think our diet increased in protein, possibly because we were better at running after our food. And in turn, all that protein was once again helpful for developing those big brains of ours. So the next time you're out for a run, or even thinking through a tough problem, remember, thank your butt. It's a big part of being human.
Maybe that was a softball to start. After all, Hank's take that butt is legs was a pretty hot one itself. We're just saying that your butt is the muscle and fat that help you walk upright, and your legs aren't. But might be one of those things that you don't define so much as you know it when you smell it. And that's why it's so important to shower every day. But Stefan will tell you that maybe that's not necessary.
Nothing really beats a steamy shower to relax after a hard workout. And good hygiene is how we protect ourselves from infections and the social stigma of being smelly. But do you really need a daily shower to stay clean, or is it doing more harm than good? It turns out there aren't any definite rules, but some scientists have recommendations based on what we know about the biology of our skin and what might be living on top of it. Your skin is a giant organ that physically blocks harmful stuff from getting inside your body. It's covered in a bustling community of bacteria, fungi, viruses, and even teeny tiny arthropods. They make up what's known as your skin microbiome. For the most part, researchers think that these microbes you carry with you are pretty harmless. But as you interact with the world, you can pick up other microbes that could make you sick. And when you rub your eyes or put food in your mouth, your hands can help those germs slip past your skin and get into your body. Which is why washing your hands with soap is so important, especially if you work in medicine or food service. See, soap is a surfactant, which means it's made of molecules that can bind to water and things like dirts, oils, or bacteria. This lets you rinse all that junk away and fewer germs means a lower chance of infection. But what about washing the rest of your body? Well, your skin has glands that are always producing sweat and oils, which cools you down and keeps your skin moisturized and healthy. But there is a fine balance. Certain bacteria break down sweat and release smelly molecules like thioalcohols. And if your skin has too much dirt, dead skin cells, or oils, your pores can get clogged, leading to problems like acne. So showering with soap can help get rid of some sweat and oil to manage your looks and smell. And in some cases, like the night before a surgery, a doctor might tell you to shower with an antimicrobial soap, which has extra chemicals to kill microbes. If a surgeon is making cuts in your skin, they don't want any potentially dangerous stuff sneaking in. On the other hand, showering too often might not be great for you either. For one, it can mess with the microbes that naturally live on your skin. And since soap and water wash away oils, your skin might get too dry so the outer layer cracks, making it easier for disease-causing microbes to slip into your body. So dermatologists seem to say that daily showers where you scrub everywhere with soap is probably overkill, unless you sweat a lot like people who work out every day. Otherwise, showering every couple days should be just fine to keep your skin microbiome healthy and happy.
If showering every few days turns your world on its head, just wait for this next hot take. It turns out that we don't know what trees are. SciShow's official stance is that trees are not a thing so much as a strategy.
Trees come in all shapes and sizes, but as different as they are, you probably know a tree when you see one. Our definition of tree is actually pretty wishy-washy, but whether you're in the tropics or a boreal forest, it's generally some tall plant with a leafy crown and a bunch of branches on a thick woody trunk. But some of the things we call trees aren't so tree-like at all. They're actually relatives of grass. And while many grass relatives get away with moonlighting as trees, there are a few telltale features that will expose these trees' grassy roots. Picture the most famous and wide-ranging of all tropical trees, the coconut palm. These trees can grow over 30 meters tall, and their strong trunks are used to build bridges and homes. But the truth is, the coconut palm is actually more closely related to the turf on your local soccer field than to any kind of woody tree. And one telltale clue is in how its seeds sprout. When the seed of a coconut palm sends up its first shoot, it has a single leaf on it. And that's important, because when we're talking about grass-like plants, we're really talking about a large group of flowering plants called monocotyledons, or monocots for short. A cotyledon is the first embryonic leaf that forms in a seed. And as the name suggests, monocots only have one of them. It's a feature that all grass relatives have in common, including plants like lilies and asparagus that don't look much like grasses but are all monocots. On the other hand, many common trees like oaks, maples, and sycamores are so-called dicotyledons, or dicots. That's because they have two of these seed leaves. But even if you're not around to witness a tree's first moments of life, there are other ways to spot a tree that's actually related to grass. And one big clue can be the trunk. Most tree trunks have an outer layer of protective bark, and underneath they have woody tissues transporting sugar and water. These trees produce a new layer of those tissues each year, which is why if you look at any woody tree stump or sawed-off branch, you'll see those signature tree rings that reveal its age. But not all trees that look woody actually are. For example, certain types of bamboo can grow into dense towering forests, and these plants can have really strong, hard trunks. But their structure is dramatically different from the trunks on most trees. Instead of growing outward by adding a new layer to the trunk each year, bamboo trunks only grow up. They're made up of a series of hollow shoots connected by nodes. So even though bamboo evolved to have a strong, thick trunk, its growth pattern is a dead giveaway that it has grassy origins. Still, some monocots have trunks that look a lot like the typical woody tree trunk, so at first glance, it's harder to pinpoint their origins. But in these cases, the roots often reveal the truth. For instance, one type of palm tree that grows in the Amazon rainforest has a trunk that looks pretty average and woody, but the roots are unusual. This tree grows large, straight roots that emerge directly from the trunk, and they're made of regular plant tissue, unlike most tree roots, which are made of a specific kind of tissue that's only used to make roots. And instead of growing and branching out deep underground like the root systems on most trees, these roots often grow higher up on the trunk and stay in shallow ground. These exposed roots can continue to transport gases and nutrients when the soil is completely waterlogged, so they help the tree cope with frequent floods. And they also point to this palm tree's grassy origins, because this type of root system is typical of monocots. Finally, if the trunk and roots don't give away a grass disguised as a tree, one other place to look for a clue is in the branches. A lot of monocot trees like bamboo and palm trees show no no real branching despite growing a tall, strong trunk. But some grassy trees have evolved to be so tree-like that it's very difficult to suss out their grassy origins at first glance. The so-called dragon blood tree, found only in Yemen, checks all the normal tree boxes. It's got a thick trunk, smooth bark, and yes, even a wide branching leafy canopy. It even has growth zones that resemble the tree rings of woody trees. But its genetics reveal that even this tree is a monocot. It has just evolved many of the same traits as ordinary trees. Which raises the question of why all these grass relatives are now living their best life as trees. And what it comes down to is the fact that being a tree has a lot of perks. For one, growing tall means better access to sunlight. Thick trunks also add strength and stability and can help plants live longer. Plus, trees are great at enduring short-term stresses like droughts and floods. And since they're so good at surviving, trees have evolved many times. They don't have one single evolutionary origin. So sometimes when you're looking at a tree, you're looking at a monocot, other times a dicot. And trees like pines and cedars are neither, but are definitely still trees. And that's simply because trees aren't a taxonomic group. It's more a loose term we apply to plants that just kind of look the same. So there are many distantly related plants that we call trees, and that includes some pretty incredible relatives of the grasses.
While we're talking about what trees are or aren't, let's get this next hot take out of the way. Trees are all dead inside.
You know what's wild? Sometimes cells don't need to be alive in order to do their job. In some kinds of tissues, cells can be completely deceased and yet still dutifully working to keep their organism alive. This is notable in plants, where trees take it to the extreme. Trees don't mind being full of dead cells. It is their way. But archaeologists hate it. Finding an ancient piece of wood can frustrate an archaeologist to no end. Here's how trees pull off living dead status and exactly what those archaeologists are so annoyed about. Trees contain a surprising amount of dead tissue. To some extent, most plants do. All higher plants have a dead tissue called xylem used to transport water around the plant. Xylem is basically just plant plumbing. It's made throughout a plant's life when long rows of special double-walled cells grow and then die, leaving their skeletons behind for the plant to use as a straw to move water and minerals through the plant. Trees and other woody plants have perfected the use of xylem. Trees are tall, and they need a way to get nutrients from the roots to the crown where the leaves and reproductive parts live. At some point in tree evolution, woody plants like trees discovered it was easiest to just make a superhighway system out of dead tubes to transport their juices from their bottoms to their tops. In addition, xylem provides mechanical support to the big, heavy tree. Xylem in the trunk provides scaffolding to keep the whole plant from drooping over. The live cells in a tree are concentrated in the outermost portion of the trunk, called the sapwood. The xylem there contains living parenchymal cells, whose business it is to do things like help the tree grow, heal wounds, and store nutrients. But in the case of most trees, the center of the trunk, the heartwood, is formed as the tree grows, and all the xylem in there is compressed and dead as a doornail. However, heartwood is incredibly strong and resilient, so it keeps the tree standing. Trees have figured it all out. They're not just surviving, they are thriving. Great for trees, right? Yet archaeologists take major umbrage with this whole approach. They're vexed to the point that they've officially named the tree way of doing things the old wood problem. I know, pretty scathing, but this is why archaeologists are beefing with trees. Archaeology deals heavily in carbon dating to create an accurate picture of when things happened long ago. Carbon dating can tell us how long ago an organism died. Carbon dating works because our planet has lots of the normal vanilla flavor of carbon called carbon-12, and it also has a fair amount of another flavor called carbon-14. These are both isotopes of carbon. Carbon-12 has 6 neutrons and 6 protons in its atomic nucleus. Carbon-14 still has 6 protons, so it's still carbon, but it has 2 extra neutrons along for the ride. Carbon-12's 6 neutrons make it stable, but carbon-14's 8 neutrons make it prone to radioactive decay. Earth's atmosphere has plenty of carbon-14, which living things are constantly absorbing into their tissue. However, this absorption immediately stops after death, and carbon-14 begins to gradually decay into other atoms at a known rate. Scientists can figure out how long something has been dead by measuring how how much carbon-14 remains in an organic object, up to as many as 60,000 years ago. You might see where I'm going with this. If you carbon date heartwood from a tree that some Neolithic lumberjack cut down to build a house, you might get a number that's hundreds of years older than the artifact or site you're trying to date, because the heartwood died before the tree did. The tree was alive and happily growing for a couple hundred years, but its heartwood wasn't taking on any more carbon-14, so the ratio's off. For archaeologists looking for clues about what folks were up to millennia ago, they're most likely to stumble on wood preserved in old fire pits. Charcoal is often well preserved because it's resistant to microbial breakdown, so it's a very reliable source of material to date. Except that the old wood problem can make it really difficult to tell when the fire actually burned, for two reasons. One is that you don't really know when the wood was cut down relative to when it was burned, but the other is because so much of a tree is dead. If the charcoal you're dating was sapwood, it's probably fine, but if it's heartwood, it was dead well before it was burned. This means that exactly when the fire pit you're looking at was last active is anybody's guess, but you're most likely to get an estimate that's a bit too old. Now that archaeologists have worked out that this is a problem, they try to find literally any other non-wood organic object to date if they possibly can. But sometimes wood is the only option, and in fairness, not all wood is bad. Some shorter-lived tree species yield more accurate carbon dating results, and wood decays faster in some environments, so it's less of a problem for archaeologists working in those parts of the world. For this reason, it's been difficult for archaeologists to get a handle on how big a problem wood actually presents to their discipline. For example, the guy who first flagged the old wood problem in the 1980s was working in southern Arizona, where there's a lot of wood in archaeological sites because it doesn't decay as quickly as it does in damp regions. But he saw it as a universal problem for archaeologists, no matter the environment they're working in, due to the variety of tree species in temperate environments that are resistant to decay. For a few decades, researchers were debating over whether to strike wood charcoal radiocarbon data from the record completely. However, recent papers have approached the topic with a little more equanimity, like the 2019 study from South Korea that found little difference between the ages of wood and soybeans from the same sites, suggesting old wood doesn't seem to pose a big problem for Korean archaeology. And there are little tricks to getting around the problem too, like selecting smaller diameter wood like twigs when possible, or using dendrochronology, a technique that matches unique tree ring patterns with known climate or historical events. In the end, archaeologists still have to work all that out. So this is all to say, when it comes to wood, archaeologists can sometimes be hundreds of years off in their dates, which probably has them feeling kind of dead inside, like a tree.
That one might have been hard to swallow because trees grow and change, so how can they be dead? Like they have behaviors that a dead organism can't have. Behaviors that make us claim trees are like salmon.
When you picture a tree, you might imagine a tall, sturdy pine or a reliable apple tree, something that produces flowers, fruit, or seeds every year, for dozens of years. And most trees are like that, but the opposite kind of trees exist as well. Much like many salmon species do in the animal world, these trees reach reproductive age, release their seeds in a single massive burst, and then promptly die. And these aren't short-lived trees either. We're talking plants that grow for decades before fruiting once and then kicking the bucket. It's hard to imagine how this strategy could help your species survive, especially when all the trees around you are putting out seeds year after year. But there is a good reason it works, especially for one species of rainforest tree. If you're an animal like a salmon, this strategy of having one big reproductive event before dying is called semelparity. But if you're a plant, it's called monocarpy. Monocarpy is more common in short-lived plants, especially ones that live in environments where growing conditions are poor or can dramatically shift, like bogs, deserts, or alpine environments. And that makes sense. These adult plants have to take advantage of a narrow window of ideal conditions, especially if they're not likely to survive until the environment is friendly again. Meanwhile, where conditions are stable, you'll rarely find monocarpic plants. And it's even more unusual for a long-lived tree to put all of its eggs in one basket, or I guess its seeds into one fruiting. Putting a ton of energy towards growing into a big woody tree and then dying after one fruiting kinda seems counterproductive. So these stable stable environments tend to have polycarpic plants, or ones that continually flower. Except the rainforests of South and Central America are home to a handful of monocarpic trees, and one of them is doing incredibly well. It's called Tacagali vesquezii, and it's so successful it might be the 10th most common tree species in the Amazon rainforests of northern Bolivia. In a 2005 paper, researchers tested how these trees were so plentiful despite reproducing just once, and they had a handful of hypotheses to investigate. For instance, previous studies had suggested that monocarpy in the genus Tacagali could be an extreme version of masting, where trees produce extra fruit to overwhelm their predators. Predators can only eat so much before they have to stop gorging themselves. So by producing way too many fruits and seeds, the tree would ensure that there are some survivors left to start their life cycle again. In fact, you can view monocarpy as masting turned up to 11. Every tree flowers and produces fruit at the same time, and then they die. Except when the researchers looked at related masting tree species in Panama, they noticed that this strategy didn't seem to make as big of a difference as suspected. Insects and other animals still ended up eating the majority of the seeds, even when released en masse. Also, although T. vesquezii does release about twice as many seeds as the average polycarp, its behavior doesn't look like masting. If this species were trying to overwhelm predators, you'd expect to see all the T. vesquezii in the forest go a long time without reproducing until suddenly, boom, they release all their seeds at once. Kind of like how periodical cicadas don't hatch for years until suddenly every tree in your neighborhood is full of screaming insects. But that's not what happens here. So another hypothesis was that trees in the genus Tacagali might be so plentiful because they have better survival rates as juveniles. And this seemed true, at least at first. The recently germinated seedlings did have a higher survival rate than similar species, specifically species whose seeds are spread by the wind. But it didn't stay this way. Once they got a little bigger, T. vesquezii's saplings ended up dropping off, and their survival rates ended up worse than those of polycarps. So there goes that hypothesis. Meanwhile, other researchers studying other trees in the genus thought that maybe it's the gap in the canopy left by the dead tree that led to T. vesquezii's success. They'd hypothesized that the adult trees die to give their offspring more access to sunlight, so that they're not forced to live in their parents' literal shadow. And while juveniles of T. vesquezii do typically occur in bright conditions more often than other tree species found around them, this doesn't explain their success. That's because, like we mentioned earlier, T. vesquezii seeds are spread by wind, so not a lot of them fall directly below the parent anyway. Whether or not there's a gap in the canopy there doesn't matter as much. So this isn't the whole story. And that brings us to a final hypothesis. This last idea was that there are so many T. vesquezii trees because they just grow faster. The thinking was that this tree can put all of its energy into growth until it reproduces. Other trees have to devote a ton of resources into reproduction every year, so they don't grow as quickly. And ding ding ding, this was the winner. The researchers learned that this tree can grow fast, with its diameter expanding about 4 times faster than its polycarp neighbors. And much like the contractor who built some of my previous apartments, this tree does this by cheaping out on construction material. Instead of putting energy into making dense wood, it produces a lot of lightweight wood and a big leafy crown to capture sunlight. As a result, it reaches reproductive maturity in only 49 years, which sounds like a super long time, but it's shaving about 30 years off the average polycarp reproductive age in these forests. This strategy also means more individual trees will reach this stage because they don't have to survive as long as others. So in the end, more of these plants get to reproduce, and the next thing you know, they're one of the most common species in this rainforest. Like the salmon that pull off an incredible journey to reach their spawning grounds after years at sea, these trees put everything they've got into reproduction. They spend decades growing to towering heights, surviving everything from weather to pests along the way, and in the end, it all culminates in one dramatic event: releasing their seeds. But even though T. vesquezii dies in this process, their adaptations allow them to carve out their own niche, even when surrounded by trees that pump out seeds year after year.
In that one way, maybe trees are like fish, but based on how many of these hot takes are about trees, clearly they're just weird. Since trees are a special case, there's no way that Hank is about to tell me anything else is like a fish. No way that the next hot take is that people are fish.
There's this funny story about beavers. In the 1700s, a Canadian bishop asked the Catholic Church if it was okay to eat beaver during Lent, a period of time when Catholics abstain from eating meat. But eating fish was okay. And look, beavers spend so much time in the water that they should totally count. And remarkably, the Church agreed. Beavers, at least as far as it mattered during Lent, were fish. Well, in most cases, people would probably say that beavers obviously should not count as fish. They have fur and live babies and breathe air, for instance. As for what science says, oh boy. See, the thing is, the answer is surprisingly complicated. Because beavers are not fish, but that might be because either we're all fish, or fish kinda don't exist. In the end, this is going to be a story about different ways of grouping animals together, why we use language the way that we do, and how evolution has made things kind of funny. For a long time, people grouped animals together by some common body plan or lifestyle, like fish, reptile, or bird. This is good because it means we can talk about many different organisms at once without needing to name each one individually. This lets us make useful general statements like, it's fish tacos for lunch today. And this is generally how science worked too. But over the years, as we learned more about evolution, it became clear that this old way of talking wouldn't work as well. There were too many exceptions, too many groups of things that seemed similar but weren't when you looked under the hood. Instead, we developed a new, more scientific way of grouping animals together called cladistics. In this approach, we sort living things into groups, or clades, that reflect their evolutionary history and how closely related they are. There's a couple of different ways to do this precisely, but a good example might be to say that rather than trying to define primates by things like opposable thumbs or big brains, instead, we would say the group of animals known as primates includes all species that are descended from the most recent common ancestor of humans, apes, and lemurs. That's a bit of a mouthful, but defining things this way is actually really useful for biologists. It gives us a way to make predictions, for instance. Like if two different species are grouped very closely together, we can predict that their genomes must be relatively similar. This runs into funny situations, though, like, again, the question of whether beavers should count as fish. Let's take, for example, the evolutionary history of a very definitely a fish, like a goldfish. Its early ancestors were some of the first vertebrates. Unlike their close, more worm-like cousins lancelets, they had a backbone, which let them swim more powerfully. As time went on, this group split. Some, which would become the ancestors of hagfish and lamprey, stayed roughly how they were, but the group that led to goldfish would go on to develop powerful jaws. Over time, more groups split off as more characteristics evolved. Sharks and rays split off, while the goldfish's ancestors got bony skeletons. In another split, lobe-finned fishes diverged. These are the ancestors of fish with thick, fleshy, paired fins for limbs, like coelacanths and lungfish. Meanwhile, our goldfish's ancestors went on to develop fins made of bony spines connected by skin. Eventually, we arrive at today and the familiar goldfish. So far, so good, and everything we mentioned so far would be in the category of what most people would call a fish. But it turns out, the beaver's evolutionary story is largely the same. Beavers share the same vertebrate ancestor as goldfish, and their ancestors also watched hagfish and sharks split off, and as lobed-finned fish split off, some of them, a group called tetrapods, used their fleshy limbs to haul themselves onto land, eventually giving rise to early mammals, and eventually to our beaver friends. And of course, we humans are in there too, in the tetrapod group, along with every other tetrapod, like frogs and alligators, snakes, birds, and horses. If you zoom out, the tetrapod branch is just one nestled among a bunch of different fishy clades. So back to the question, are beavers fish? Remember that in the cladistic approach, for a definition to be useful, we want to include everything that is descended from a common ancestor so that we can make predictions about who is related to whom on an evolutionary scale. So this means that if we want to be able to say that sharks, hagfish, and goldfish are all fish, we kind of have three options. One, animals like humans and beavers are fish. Two, you have to carve out some ridiculous definition, like fish would be any organism that is descended from the most recent common ancestor of both hagfish and goldfish, except for the subset of that group that is descended from the most recent common ancestor of frogs and beavers. Which is awkward, but it also kind of defeats the purpose of cladistics. Or three, fish, as a scientific category, don't exist. So, in the end, Are we fish? Well, we are all vertebrates, and there are smaller categories like ray-finned fish or sharks and rays, but there is no single fish category in the cladistic approach. If that sounds like scientists being unnecessarily pedantic, they do it for a reason. Despite the foibles, the cladistic approach has been a huge success. It helps scientists talk about the evolution of life on Earth in a way that makes sense. And if there are some weird edge cases, it's not the fault of the system, but an opportunity to marvel at the history of life on Earth. And it also means that we use language in different ways based on what is useful for that conversation. That means it's still okay to call a fish, and if it's Lent, maybe even to include beavers in that category.
I guess we're always learning new things about ourselves, but that's a great thing about being human. We're lifelong learners, and that probably helps boost the old IQ. Or maybe not, because the next hot take is that IQs don't really measure intelligence at all.
When people talk about smarts, intelligence quotient, or IQ, always seems to come up. People love to bring up how Einstein had a genius-level IQ of 160, and to join Mensa, you need to have an IQ of at least 130. But is IQ even a good way to measure intelligence? Well, that depends on how you define intelligence. IQ scores may be a useful shorthand to talk about education strategies for big groups of people, like when discussing public policy. But IQ can be affected by a lot of factors, even things as subjective as your motivation while taking the test. The first sort of IQ test was invented by the French psychologist Alfred Binet in the early 1900s. A law in 1882 aimed at egalitarianism said that any healthy child had to go to school and learn the basics, like reading, writing, arithmetic, history, public policy, and the natural sciences. The law even included a special consideration for children with disabilities, like deafness or blindness. But the French government acknowledged that not every kid would be able to keep up with a normal curriculum, for lots of possible reasons. So Binet and other psychologists were commissioned to create a standardized test to measure how different kids handled their schoolwork. Along with Théodore Simon, Binet developed the Binet-Simon test, in which children would answer a series of questions until they couldn't anymore. That way, kids could be grouped in classes with students with similar scores, instead of relying on their age or the subjective judgments of teachers. In the next decade or so, this scale was revised for use with both kids and adults and renamed the Stanford-Binet Intelligence Scales. This popular IQ test is still used today, along with other standardized tests that are meant to measure learning ability, sometimes defined as how quickly and easily we learn new things. On early versions of the test, IQ was calculated by taking a person's score on a standardized test, dividing it by their chronological age, and then multiplying the result by 100. In more modern versions, you're basically ranked against other test takers. The scores of a group of people are scaled so that 100 is the average, and your IQ score is determined based on where you are in relation to that average. But here's the thing, whether or not IQ tests actually measure your intelligence depends on how you're defining intelligence. In simple definitions, intelligence is the ability to learn new things or adapt to new situations. But the definition could also include the ability to use logic or reason or to think abstractly. These definitions are all focused on intellectual capacity, which is how intelligence is defined by the American Psychological Association. And they don't include other kinds of intelligence, like social or emotional intelligence, or things like creativity or self-awareness. The Stanford-Binet test, for instance, focuses on testing 5 main categories of information: baseline knowledge, basic mathematics, visualizing objects in space, working memory, and fluid reasoning, or the ability to solve new problems. So depending on what you're trying to understand about someone, IQ tests might be useful, or they might be a waste of time. It turns out that your IQ score can be affected by a lot of different things, and because intelligence is so complex, we're not sure how strongly different factors might affect it. There's some evidence that says cognitive abilities are somewhat heritable, meaning there might be some kind of genetic component to IQ, but it's not that simple. Recent studies have shown that IQ tests are affected by motivation. For example, one 2011 meta-analysis found that people who are offered cash if they do well on an IQ test scored higher than people who weren't offered anything, like up to 20 points higher for just a $10 reward. That is a huge effect. And we know that motivation can play a role in other things, like your grades and your career path, that could be wrongly chalked up to just an IQ score. IQ also seems to be affected by environmental factors. Cultural values can influence your IQ scores. For example, a kid who grows up in a community that prizes storytelling might do better on verbal sections of the test or problems that require you to remember and reuse information. How much education you get and the quality of that education may have an effect too. Kids who miss school because it's hard for them to get there, or who attend schools without many resources, tend to score lower than their peers. Even your family environment can affect your IQ, like whether you grow up in a low-income household or whether you experience a lot of trauma as a kid. So like a lot of things, IQ seems to result from a mix of nature and nurture. There are just so many factors that affect your learning ability as you grow up, from the environment you develop in before you're born to things like education opportunities and family dynamics. But psychologists seem to agree that one thing that seems to help people with learning and academic achievement is thinking about intelligence as a thing that can change. So IQ tests aren't anywhere near perfect or comprehensive, but they can help us predict how people might learn in the near future, which can make a difference in the support they receive. For instance, IQ scores can affect diagnosis of intellectual disability, which can inform public policy about education programs to support different students. It's understandable why it's valuable to have a standard way to sort of measure intelligence, like when it comes to making these general policy decisions. But it's also easy to see why IQ tests have been surrounded in controversy too. There's a lot we don't understand about intelligence, and a lot that an IQ score can't tell us about a person or groups of people. So while IQ can be a useful shorthand in some cases, it is not something that is set in stone, and do not let a number define you.
So IQ tests might not be able to detect if you're one of those people working with the last 2 brain cells. But even if you only had 2 brain cells, it would be one more than the dog Hank's about to talk about. The next hot take is that a dog can be a single cell.
The title of this video does not make sense. There are no single-celled dogs. Dogs aren't just multicellular, they're vertebrates, mammals even. A single-celled organism is by definition not a dog. And yet, between 6 and 11,000 years ago, there was a dog. I do not know the dog's name. I don't even know if it was male or female, but we do know quite a bit about it. It was black or brown with more than a bit of coyote ancestry, and it came from an inbred population. And also, we know it had cancer. And to understand what happened next and how truly bizarre it is, you have to understand what cancer actually is. And I know what you're saying, Hank, I know what cancer is. And maybe you think you do. You think that cancer is a disease caused by an organism's cells malfunctioning and reproducing uncontrollably and moving to different parts of that organism's body. And that is true, but also it's kind of not true. The question is, are the cells malfunctioning or are they succeeding? And to answer that question, you have to ask another one. Why do malfunctioning cells so often reproduce uncontrollably? Now, it's convenient to talk about evolution in terms of what it wants. But evolution doesn't want anything. One school of thought proposes that natural selection is simply successful traits and their associated genes succeeding and being passed on to future generations. And nearly every cell in a dog is trying to help the dog survive. This is what evolution is selecting for at the scale of the dog. Cells work together to keep the animal functioning so that the genetic material can persist in the current dog, but more than that, in the offspring of the dog. But sexual reproduction isn't the only kind of reproduction. Single-celled organisms often reproduce asexually, and natural selection, nonetheless, still results in evolution in those species, just more slowly. And we can imagine that inside of a dog, natural selection isn't just selecting on the scale of the dog, it is also selecting on the scale of the cell. Natural selection is not a complicated idea. If something can make more of itself, then there will be more of it. If a rat has a gene that allows it to have 1,000 babies, then that gene will spread throughout the rat population pretty dang fast. But if a cell inside an organism has a gene that allows it to have 100,000 or 100 million copies of itself, that gene will also spread through the organism very, very rapidly unless something stops it. And when that happens, when individual cells inside an organism evolve ways to replicate themselves unchecked, we call that cancer. One way of thinking about cancer is that it is simply the result of evolution acting on the cells of a multicellular organism rather than the organism itself. Now, this is kind of a hot take, but it's one that makes sense according to what we know. Cancer cells are evolutionarily successful, not for the organism, of course, but for the cell. Seen through this lens, cancer is not an organism's cells malfunctioning. It's kinda a return to a single-celled lifestyle. A step, at least. They still require you as a host organism, and since they usually hurt the host, you in the process, it's not a great step. Still, your cells have become more like a pathogen. They have human DNA, but they have started living for themselves instead of for you, metabolizing, replicating, and evolving. So one way to look at cancer is that it is a single-celled organism with a multicellular organism's DNA. Now, evolutionary pressure on the organism level encourages lots of systems for preventing individual cells from growing unchecked, because when that happens, the organism can no longer pass on its genes because of how it is dead. But evolution at the level of the cell is hard to overcome. Natural selection is always selecting. Genes are always mutating. It only takes one cell being really good at evading the immune system and selfishly replicating itself perpetually for the whole organism to crash. Great, Hank, thanks, wonderful. That's all very terrifying and upsetting. Cancer is the most natural thing ever, and despite the fact that our bodies have evolved a tremendous arsenal of tools to prevent it from killing us, it nonetheless remains an extremely common cause of death. But for the most part, all of this is just semantic line shifting. Is cancer a kind of single-celled organism? Is a hot dog a sandwich? It doesn't really matter. It's a deadly disease, and it's not like it's gonna jump off of a sick person and go live on its own. Unless it does exactly that. Remember that dog that died thousands of years ago? Well, before it died, its cancer managed to spread to at least one other dog, potentially through sexual contact. These cancerous cells were now a contagious pathogen that for thousands of years have been successfully living the single-celled lifestyle. This disease is called canine transmissible venereal tumor, or CTVT, and you really cannot say that the cells that cause it aren't alive, because they grow, they reproduce, they metabolize, and they evolve. A dog with CTVT does not have a cancer made of its own cells. It has a disease made of cells that are descended from a dog that has been dead for thousands of years. As the authors of a 2014 paper put it is remarkable that a somatic genome whose DNA would normally have survived for no more than 15 years during the life of one dog has continued to exist for several millennia as a parasitic life form. CTVT's survival and global dominance is a testament to the ability of the mammalian somatic cell genome to adapt and persist in a new ecological niche. In other words, this cancer, if you look at it a certain way, is a single-celled dog. Now, the success of CTVT is very weird and unusual. Cancer cells evolve to succeed inside a very particular environment, the body they are part of. And they evolve to evade the immune system of that one organism. That's why cancer jumping from one organism to another is extremely rare. Now, we're not entirely sure how CTVT did this, but other transmissible cancers have occurred in species that were in population bottlenecks, which just means that their population shrank a lot and the individuals in that group wound up being severely inbred. And it isn't difficult to imagine this happening to a population of early dogs. In a situation like this, CTVT would be able to jump from dog to dog because their low genetic diversity might have made it less likely that their immune systems would notice that those cells belonged to somebody else. Given the new evolutionary pressure not just to grow in one organism, but to be transmissible and survive in other organisms, CTVT continued to evolve, and it can now infect not just most dogs, but also related animals like jackals and coyotes. The fact that there is a line of pathogenic cells reproducing asexually descended from an ancient dog that has persisted for millennia is one of the weirdest things that I have ever found out about biology. And it only makes sense to me if you view cancer not as a malfunction, but as a kind of terrible evolutionary success. Cancer, through this lens, is a kind of biological inevitability, and so we need to see it as a thing that our bodies have developed systems to control, and that we can help our bodies control. Evolution allows these selfish cells to evade both our body systems for destroying them and our technologies for destroying them. We are in a race, but it is one that we are winning more now than we ever have before.
At this point in the compilation, I wouldn't blame you for second-guessing your grasp on reality. A dog can be one cell big, all trees are dead. I'm a fish. Next thing you know, Hank will tell you that you're not supposed to put medicine in medicine cabinets.
Does your bathroom have a medicine cabinet? Do you keep medicines in it? Because, fun fact, this basic architectural reality of many homes invites us to store our medications in the worst possible place. Heat and humidity are the enemies of most common medications. Luckily, most of the time the consequences are mild. Except for that one time when it might have been really bad. Like, really bad. We'll get there. If you keep your medicine in the bathroom, you are not alone. I certainly do. Maybe it helps you remember to take your meds if you see them every time you brush your teeth, or maybe it's just the most convenient place. But bathrooms are not the best place to store medicine. Lots of drugs degrade when they are exposed to light, heat, or humidity. The light part is actually why medicine bottles are like brown colored, so that not all the light gets through. It's like they have sunglasses on. If anybody in your household, takes hot showers, the chances are your bathroom hits at least one of those criteria. Now, it's not like scientists are not aware of this problem. It is a stunningly dull job, but there are people who monitor the long-term stability of medicines before and after they hit the market. Basically, you stick the medicine in these big chambers meant to mimic the conditions of your average Florida afternoon, like some dark, twisted reflection of a refrigerator. Then you pull some of it out every now and again to make sure it's all still there, or if it started to degrade. And while yes, this is a boring job, I used to work in a quality control laboratory, somebody has to do it because we want our vitamins and pain relievers to still work even if the AC goes out for a few days. This information is where expiration dates come from. And what we learn from these kinds of experiments is that storing your medicine under too warm or too humid conditions is usually not the end of the world. Your medicine might go bad before the expiration date, or it might lose a bit of its potency, but still work okay. Take insulin, for example. Insulin becomes less effective when it's stored at temperatures above 30 degrees Celsius, but some sources say that you can still use it in an emergency. Lidocaine also remains stable after 6 months at 40 degrees Celsius. Most over-the-counter medications are designed to be stable at or close to room temperature, but you cannot exactly guess based on the kind of medication. Consider 4 common pain relievers: acetaminophen, aka Tylenol, ibuprofen, aka Advil, naproxen, aka Aleve, and aspirin, aka aspirin. You'd think that all of these would be the same, especially Advil, Aleve, and aspirin, because they all belong to the same class of drug, and they all start with A. And yet, Advil and Tylenol work best when they are kept between 20 and 25 degrees Celsius. Meanwhile, aspirin and Aleve are a little hardier and can tolerate temperatures between 15 and 30 degrees. And that's just regular tablets before you get into stuff like liquid capsules and stuff. I know. Just check the label, ask your pharmacist, don't go based on vibes. Sometimes improper storage conditions can have mild downsides. For example, consider aspirin again. Aspirin slowly breaks down into 2 other compounds, acetate and salicylic acid. Acetate plus water makes acetic acid, otherwise known as vinegar, which will not hurt anything in small amounts, but also will not do much for your headache. Salicylic acid might still offer some pain relief. Ancient Sumerians extracted it from willow bark and used it as one of the very first painkillers, but it can be hard on your stomach. We also could not confirm whether or not it works as a blood thinner if you're on an aspirin regimen for your heart. So, if you open your aspirin bottle and get a whiff of vinegar, probably time to toss it out. But every once in a while, storing medicine improperly turns out to be dangerous, which means we need to talk about ranitidine. Ranitidine, which was sold under the brand name Zantac, is a heartburn medicine. If you're a heartburn type of person, you may have noticed that Zantac disappeared from shelves back in 2020. That's because the FDA was informed that a chemical called N-nitrosodimethylamine, or NDMA, builds up over time in medicines that contain ranitidine. N-NDMA is classified as a probable human carcinogen, a substance that might cause cancer. It also occurs naturally at low concentrations in water and foods like grilled meat. At low levels, it's not a cause for concern, but at higher concentrations, it could increase your risk of developing cancer. So just so we're clear though, you, the consumer, are not responsible for making sure your medicine doesn't decay into a carcinogen, even if you do keep it next to the shower. Regulatory agencies like the FDA exist to make sure medicines are safe, and the FDA FDA launched a full investigation once they learned this might occur. The FDA found that NDMA will build up in ranitidine medicines over time regardless of temperature. But NDMA accumulates faster when ranitidine is stored at higher temperatures, including temperatures that it might encounter en route to pharmacies or when it is stored at home. Several companies issued a voluntary recall on ranitidine while the FDA investigated the issue in 2019. Then, in April 2020, the FDA asked manufacturers to pull ranitidine off the market. Regulatory agencies in the European Union and Australia followed soon after. Regulators found that the amount of NDMA that builds up in ranitidine products could exceed the acceptable daily intake limit, the amount that has been shown to cause adverse effects. For NDMA, that's about 96 billionths of a gram per day. There is some good news, though. The FDA pulled ranitidine to be on the safe side, and a 2023 study found that ranitidine use was not associated with higher cancer rates. Now that could change, but there's no reason to scare you if we don't know. Plus, NDMA doesn't build up in other heartburn medicines like Prilosec and Nexium, so there are other options for managing heartburn. Also, Zantac turns out is back on the market. It's just a different drug with the same brand name, so it should be all right. Zantac people, please don't sue us. Again, even if you kept your Zantac in the hottest, muggiest, most swamp-like bathroom in your house, it's not your fault that this happens. We included it because it's like the exception that proves the rule. Usually, if your meds get a little toasty, they are fine. And we know that because there was a time that it wasn't fine and the system in place caught it and stopped it. If you would like to be extra sure you are storing your medicine safely, here are some steps you can take. Keep your medicine in a cool, dark, and dry place out of reach of children and pets. Depending on your situation, a closet, dresser drawer, or kitchen cabinet away from the stove might be good choices. If there isn't anywhere that works other than the bathroom, run that fan during and after showers to keep the humidity low. Also, take out any cotton balls that come with your medicine bottle. Cotton soaks up moisture from the air, and that water then gets trapped in the bottle and can break down medicines over time. It's there in the first place so that the pills don't rattle around and break. So you don't need it anymore once it's at home. Dispose of any medicines that have changed color, consistency, or smell, and check with your pharmacist for instructions on how to safely get rid of old medicines. Sometimes they'll even let you drop off certain pills with them. And also, if you are still hanging on to any old Zantac or other medicine with ranitidine, it's time to throw that out and ask your doctor about alternatives. It's probably a good thing that most medicines don't turn into anything harmful when you store them under less than perfect conditions, because humans are often less than perfect. I know I am. That's just how we are.
It's almost a relief that there are no more videos in this compilation. You can only take so many hot takes at one time. So let us know your own science hot takes once the shock has worn off.